European bat lyssavirus 2, a bat biologist in Scotland, where they don't have any classical rabies virus, died with the symptoms of rabies, basically diagnosed as having rabies, and would have been protected had he been vaccinated against rabies. And also of interest is the vast majority of these are bat viruses. So rabies virus, as we know it, has many different hosts, but it is originally a bat virus. So I guess somewhat like COVID appears to be, these are things given to us by the bat population. So next, please. So these are the bat lyssaviruses, and you can see the rest of the world, well, the old world, has many of these different lyssaviruses, whereas North and South America only have classical rabies virus in the bats. Next, please. And if we look at terrestrial mammals, we still are limited in North and South America to the classical rabies virus, RABV. There are multiple different terrestrial hosts. This becomes important. So we'll go to the next one now. If you look at North America, we can see the distribution of classical rabies virus through the various hosts that spread it. In our province, we're primarily concerned with Arctic fox rabies, which, as you can see by the red, sort of rings the Arctic coastline. The fact that it's showing it on the west coast of Newfoundland is a historical thing. This does not necessarily mean it is there on a permanent basis. These other fox variants that you see, north central skunk, raccoon, whatever, are the plagues that are visited upon other people further south. So raccoon rabies, as an example, started in southern Florida in the 60s or 70s and has moved completely up the east coast of North America, crossing over into Canada numerous times, causing a lot of concern and millions of dollars to control it. So these are the terrestrial variants, and we can go to the next slide, please. And so Arctic fox rabies, that which affects us, affects all across the Arctic circle. So it's not just the Arctic coastline of North America. Greenland and Eurasia are also exposed. And here's two photos, one of a red fox that died of rabies in Makhovik, has porcupine quills in his face. And then there's a rabid Arctic fox attacking a dog at a meteorological station off of Norway. So that's a bit of the reality that these people face. Next, please. So when we see rabies in Labrador, it is usually at a 1-year lag behind that which is seen in northern Quebec. As we'll see, northern Quebec and Labrador don't have rabies on a permanent basis as far as we know. It comes down from Nunavut. And when it comes from Nunavut, it goes into northern Quebec first and then moves across to us. So this is apparently a pattern. As you can see, in the years 2000 to 2010, the pattern sort of broke up. We continued the dotted line in Labrador to see a sort of a 4-year cycle, but it wasn't really mirrored in northern Quebec. Part of this can be related to surveillance effort. We have always encouraged that every potentially rabid animal be submitted for testing, whether it was in contact with a person or a domestic animal or not. So a fox found dead somewhere under suspicious circumstances, we would bring it in and test it. So that would give us a lot of data. If northern Quebec limits its testing, since it is expensive, it requires training and shipment, dangerous goods of a head from remote areas, since there are costs, there could be periods when there are cutbacks on submissions and you get less than you might hope for if you are interested in surveillance. So, but we do think that there's about a 4-year pattern that is preceded in northern Quebec before it goes into Labrador. Next, please. So as a bit of a history, and the history is important because it shows us where we are in terms of research and the value of research. So originally rabies was diagnosed just through thin sections of brain tissue that was stained with some of the classical stains. H&E was one of them. And so you were interpreting the presence of this virus by the presence of these inclusion bodies in the cell of here an infected neuron. That was first developed in 1903 and became the standard, but it was not excessively accurate. 50 to 80% of cases were diagnosed, or positive cases were true positives. So when at the beginning of my career, or when I went to vet school, this is the way it was commonly done. And to confirm it, particularly if there's human contact, if a fox bit a person, you'd want to make sure you knew whether that fox was rabid or not. They would take brain tissue from the fox and inject it into the brains of mice, then sacrifice the mice after a certain number of days and look for the presence of the virus there. Later on, cell cultures were used to grow up virus to try to confirm. Later on, 1965, and later, fluorescent antibodies became the norm, which increased sensitivity and are still the standard test now, direct fluorescent antibody for the presence of rabies in brain tissue. Next, please. But that was only saying the rabies virus, yes or no. We didn't know anything about the variants. Rabies was just rabies. So if you had a bat with rabies, you had a fox with rabies, it was always just considered to be rabies. But in the 1980s, with monoclonal antibodies becoming the norm, it was possible to develop these antigenic groups that you can see on the right-hand side of this table. So then we're starting to see rabies virus splitting into hosts. We have Canadian Arctic, which is the Arctic fox strain, southeastern Georgian Bay, I think that was a skunk strain, then south mid-central. And these B1, 2, 3, 4 are different bat strains. And down the bottom, those are vaccine strains. So now you can start to see the ability to determine the host of animal that was responsible for the virus. And this may appear not to be extremely valuable. If a fox bites a dog, why is a question of where the virus came from. But this becomes extremely important, as we will see in subsequent slides. So if you could move on. Next one, Tom. So here is a summary of the cases of rabies that have been confirmed in this province from 1954 to 2011, when these maps were generated. So you can see many cases in Labrador and a few times when it made it onto the island. Twice we have confirmed the presence of rabies or the presence of bat variant. So one was in a bat in Cartwright, the more northern one, and then on the south coast of the island of Newfoundland, we found a case of bat rabies in a fox. So this is very important because generally rabies dead ends in species outside of its host range. So fox rabies is fundamentally a fox disease. If a wolf is bitten, the wolf is likely able to transmit it. If a dog is bitten, it is likely able to transmit it, but this is a fairly closely related group of species. If a bat bites a fox, the fox will die of rabies, but the fox is not able to spread it. The virus is not sufficiently adapted for the fox as a host. So again, this becomes extremely important. Next slide, please. Here we can see, I'll get back to the bat in a second. Twice knowingly, this disease has crossed over, presumably from Labrador onto the island. In the left we have 1988, when we knew it had crossed over onto the island because we had sufficient or significant ice bridges from southern Labrador onto the island. And it was typical, anybody who's lived here any number of years knows that polar bears walk across on the ice. And so there's a clear path for rabid animals to move over if the conditions are right. In the right-hand map, 2003, we actually had a case in December of 2002. We'll get into that a little bit more. That started in December in basically Grossmore National Park. So we'll get to that more. Could you go to the next one, please? So here is the 1988 outbreak. So we knew this came across on the ice. First case was up in Roddickton, very close to where an animal could step across, and we had a cluster of cases around there. There was a case in Triton, but this was a fox found dead floating in the water. Associated with that, in January of the next year, as you can see on the right-hand side, in Grand Bret on the south coast, a fox was found dead on a snowbank. The person who found it knew that we were interested in dead foxes because we were paying a bounty for foxes that were trapped and killed in the target area. And he contacted us and we tested it and it came back positive for rabies. And the first thing you get from the lab is yes or no rabies. So this was a great concern for us because at that point we thought that we had lost control of the disease and it had escaped from the northern peninsula and moved all the way down to the south coast without us knowing it, because people familiar with the geography of this province, once you cross the highway around Deer Lake and go into the center of the core of the island, population density drops significantly and something could happen there and you'd never know. Fortunately, that fox came back to have died of that variant of rabies virus. So we could completely ignore it. It was an oddity. It wouldn't spread from there. We could turn off the helicopters and go back to dealing with the outbreak on the Northern Peninsula. So once we had variant typing, and you see that's in 1989, so that's just the decade that this was developed, we were able to customize our reaction based on what the variant was. Next, please. So here's an example from 2020 of the ice fields that are connected between Labrador and the island of Newfoundland, and you can see how easy it would be for an animal to walk across there. We can see in the bottom right image, there's a polar bear walking on ice being followed by an Arctic fox. So they often scavenge behind polar bears when a polar bear makes a seal kill. When a polar bear crosses, it's quite visible. When an Arctic fox crosses, it can just slip into the woods and not be seen. Next, please. So right here, we have the second outbreak on the island during my career. And the point of going through this history is to show how much more information we can get through the perseverance of research over time. So in the case on the left, we are convinced that the rabid fox would have crossed over on ice bridges. The first case was in March, plenty of ice around. The map on the right, this was 2002. Our first case was in December. There are no ice bridges. The first case was in St. Paul's, 250 kilometers south of the tip of the Northern Peninsula. So, and during our eradication of this outbreak, we could see that the cases were spreading out from there. It had not existed on the Northern Peninsula for a long time period. And we had just found, the 10th or the 15th case. We found pretty close to the first case of the disease. So this left us wondering where it came from. Why December? so far south. The closest cases we had historically had been on the coast of Labrador in the previous April, May of 2002, but nothing in between there. So just for proximity purposes, we said, well, maybe it's associated with cases in Cartwright, but it didn't make a lot of sense. But that's all we had to go on. So next slide, please. So this is when we get into the genetic works, and one of our co-authors on the paper that Don will be discussing more in depth, Susan Adden Davis, did partial sequencing of the P gene and identified 4 different clades. And you can see the circle, our outbreak on the island is not closely related to the samples from Cartwright. So we said, well, we cannot associate it with Cartwright. It had to come from somewhere else, but where, we don't know. Next, please. Looking at those, the clade that the Newfoundland samples or the outbreak samples came from, it could not be distinguished from virus isolates from all these other red dots. So there had been cases in previous years in Nain that were the same virus based on partial sequencing of the P gene alone. So, you know, there was, all we could do was conjecture at that time. You know, maybe somebody working in the north had a dog that got bitten by a rabid fox. They came back to the island, the dog got free and mixed in with foxes. But where could it have come from? No idea. Next, please. So when we look at these outbreaks, and this is just the years 2011 to 2013, a fairly significant outbreak. We have 3 different species here, as you can see: white stars, Arctic fox; brown stars, dogs; and red stars, red fox. We look at this outbreak and we look at the timings, what day it happened or what month it happened. And we basically think that it comes down from the north, but it's such a huge area and we only see cases where people congregate. So the vast majority, probably 99% of that area, we have no surveillance whatsoever in. So that just you look at this map and you come up with questions. And next, please. How are all these cases connected? We have outbreaks, but how are they connected? Next, please. It'll take 2 clicks. Okay, we see, just for example, in our province, we could have cases in Labrador City, and then within 2 weeks, cases in Nain, 500 kilometers away. How are they connected? Why do we see these things happen within a short time period when there's such a vast area? Next, please. What pathways do they take? How do the animals move? You can guess, but how can we figure that out? Next, please. And what happens in between these outbreaks? So we have these 4-year peaks. What happens in between? Does it exist in the woods and it just comes out later? Does it kill itself off and we just wait for another wave to come down? So these are the questions that remain. The more we know, the more we want to know. Next, please. So this is just my imagining, looking at it and saying, well, it's coming down from the north. Here are these north cases. And when I draw an arrow between 2 points, that doesn't mean the animal moved. I'm just connecting points. And assuming that coastline, well, animals probably move along the coastline. It's an easier pathway and there's communities that they can scavenge food from. And then, you know, coming down the coast of Labrador, but how does it get to Labrador City? Does it come down from Kuujjuaq? Maybe it comes across from Umiujaq. You know, it's just guesswork on my part to assume that. And the case in 2013 in Kashechewan is actually very significant because they do not have a history of rabies on the west coast of James Bay like that. So that could be a significant predictor for further outbreaks, but that doesn't really affect us. That's further away, but it's still interesting. Movement of rabies. So these are the questions that we're left with, and my side of this was mostly the practical side of diagnosis and vaccination and trying to anticipate what would happen and react to it when it did happen. So next. This brings us to the research, and I will have a couple of comments at the end, but pass it to Dawn now for the hard part.
Well, I hope I can summarize this well. A lot of this wasn't my work, but in any event, what I'm going to do is present the findings from this article that we now have in revision for PLOS One. The work led by Dr. Susan Madden Davis of the National Reference Center for Rabies at the CFIA, co-authored by Dr. Whitney and myself, and 2 students were involved in the project as well. Emily Fowler-Doe, who was an honor student at the University of Ottawa, supervised by Dr. Madden Davis, and Alex Flynn, who was an NSERC USRA in my laboratory. And so, as you can have, I guess, gotten the idea now from the nice introduction by Dr. Whitney, the idea of this study was that rabies virus spreads in the Canadian Arctic in both red and Arctic foxes, with Arctic foxes considered the principal reservoir host, but red foxes important in disease maintenance. And control measures sort of remain difficult or impractical for these large northern regions due to the geographic extent of the fox species throughout the region and the sort of isolated and dispersed nature of human communities and sort of resulting limitations to surveillance. So we thought we would, the objective was that we would hope to sort of shed light onto the transmission patterns of the virus by a comprehensive and comparative analysis looking at both the host and the virus population genetic structure, combined with historical surveillance data. And, you know, as hopefully you'll see by the end of my summary here of this paper, the results support the primary role of the Arctic fox in maintaining and rapidly spreading distinct subtypes of the rabies virus, while in Eastern Canada, we see that there are somewhat distinctive red fox populations that spread the virus southward via 2 independent pathways or corridors. All right, so starting with the virus work, and again, this is the work of Dr. Madden Davis, so I'm just trying to summarize it here. The first goal was to acquire a picture of the diversity of the Arctic rabies virus lineage circulating in northern regions. And this was done by sequencing the N or nucleoprotein gene of the virus. So you can see down here, just one gene. And so about 1,350 nucleotides of this gene. And it was from 278 rabies positive samples that had been compiled throughout 1977 to 2017. And within this data set are 210 Canadian samples, as well as samples from Siberia, Greenland, and Alaska. And on the right here, you see a maximum likelihood phylogeny. And what this does is this divides the rabies virus lineage into 4 to 5 sublineages, depending on how you look at it. So this is basically consistent with sort of 4 previously known sublineages of the Arctic rabies lineage. And these are A4, which primarily circulates in Alaska. Yeah, so we had 27 members of the A4 clade between 1989 and 2008, all from Alaska. And then there's A2, 15 samples collected in the 1980s and 1990s from Siberia, and 8 from red and Arctic foxes from Alaska from 1988 to 2008, and then 2 samples from the northwest region of Canada. And then A1, which is this one here that you see down here, the first one, this had sort of formerly been one lineage, all of which are from Ontario. But with this new analysis, this got subdivided into two distinct lineages, geographically distinct as well, Western Ontario and Eastern Ontario. And these, this is sort of consistent with previous work that had identified distinct Ontario viral subtypes that had evolved independently of other members of the Arctic lineage, following incursion of rabies from northern Canada in the 1950s, and with some evidence that the virus which persisted in Eastern Ontario represented a separate wave of infection. So the bulk of the samples, some 213 of them, are in this large, geographically widespread A3 sublineage. And so we'll go on to talk about that now in more detail. But notably, cases in both red and Arctic foxes are distributed throughout this phylogeny, with the exception of the A1 clade, which is found outside of Arctic fox territory. And yeah, but otherwise, and then, so most of the cases are in red and Arctic foxes, but there are some cases of dogs, wolves, one case in a human, one case in a polar bear, one case in a cat, one case in a sheep, and several cases, I think, in striped skunks throughout this phylogeny. And so notably, only the A3 sublineage is currently circulating in Canada. And so we go on to look at this in more detail going forward. And so to further explore the substructure, specifically, particularly of this A3 lineage, sublineage, whole genome sequencing of 208 samples was performed. And this is all sort of summarized now in a tree, but you can imagine this was simply a massive amount of work on the part of Susan and the CFIA and the people working with her. So the end result here is this time-scaled phylogeny generated using the program BEAST, Bayesian Evolutionary Analysis, sampling trees. And the clade itself, the tree itself is what's called a maximum clade credibility tree. And it's a time-scale tree. And so we can actually get dates on the emergence of these different subgroups within this lineage. And so based on this, we could see that the Arctic lineage itself evolved some 82.5 years prior to the most recent sample in the analysis. So that places that at 1935. And interestingly here, we see that the Ontario samples actually all group together, so differently from the previous tree. And they emerged in 1951 with subsequent separation into the West and East groups. And the A3 and A4 sublineages diverged from each other in 1955, give or take. And all A3 sublineage viruses evolved from a progenitor dated to 1975 and has since evolved into multiple distinct groups, as you can see here, which are now designated A3 1 to 18. So 1, 2, 3, etc. And with further diversification occurring in the most recently emerged groups, which are A3 17 and 18 down here. So there are well-supported subgroups within this very recent emergence of these two groups here. So what we've done, or what Susan did, painstaking work, was to map the coordinates of each case over 3 maps, over 3 different time periods. And so that's what we're going to go through in the next couple of slides. And this allows us to look at each of these sublineages, A3, 1 to 18, when they emerged and where they spread and when they died out. So this is just some summary points kind of this whole thing right now. So we only had one case from A3-1 So that's not included, and that's fine. The samples are primarily red and Arctic foxes, 70 red and 70 Arctic, with 33 other hosts, as mentioned. And overall, there's just what seems to be a complex disease transmission pattern. And only a few groups were recovered in the 1990s. Additional groups emerged in the early years of the 21st century. And most of the viral groups have not been recovered recently in Canada. And since 2010, all cases examined represent 4 main groups: A3, 2, and 8, and then 17 and 18, those 2 big recently diverged groups. And those 2 have become the prominent groups in recent decades. And we have 2 groups in that A3 sublineage, A33 and A315, which are restricted to Greenland. So we'll just kind of go through this now, looking at it group by group. And I may just have to skip through some of this quickly. So group A32, which is this group here, was recovered from a single red fox in Alaska in the 1990s and subsequently circulated primarily in Arctic foxes in the islands of the high Arctic throughout the 2000s, and continues to be recovered from the high Arctic throughout the most recent decades, but has also spread south into mainland Canada, where it's caused several cases in both Arctic foxes and dogs. You may recognize that dog, sort of around the Hudson Bay area there. Now, A3-3, the next evolved group, was first isolated from the northwest coast of Greenland in the 1990s. And since then has spread as far as the southern parts of Greenland, with cases from several West Coast communities, and has not been recovered from Canada, but it's thought that this group emerged from A32 viruses that were circulating in the northeastern Arctic region and probably entered Greenland by foxes moving over pack ice from Ellesmere Island. All right, so A3-4, our next group, circulated across northern Canada throughout the 1990s, mostly in Arctic foxes, with cases on Baffin and Southampton Islands and in northern coastal communities. And then moved south into the red fox population in Ontario and Labrador during the 2000s, but has since disappeared. So that group has disappeared. A35 was first identified in 1993 in an Arctic fox on the northern tip of Baffin Island. I may not have that put in there. Anyway, and a few years later was found at opposite ends of the country in both the Northwest Territories. Oh yeah, so that's the Arctic fox, Baffin Island. Then the Northwest Territories and Labrador. So just completely far apart. And this group, A35, and its offshoot, A36, A36 circulated widely in the north and spread into Ontario and Quebec, as you can see here. Both have now disappeared before the end of the 1990s. And A37, except for one case in Quebec, was recovered exclusively in the region of Labrador, you can see here, where it caused a significant outbreak in red foxes beginning in the 1990s and continuing into the 2000s, with the last case recorded in a dog in 2004 in Davis Inlet. Now, A38 is estimated to have emerged in 2003 from that timescale phylogeny, but was first reported in 2005, at which time it was already circulating across the northern mainland, down the western shore of Hudson Bay, and on Baffin Island, and with cases in both red and Arctic foxes. And it was also recovered from Arctic foxes in Alaska. We don't have exact coordinates for those. and in Greenland. And thereafter continued to circulate with cases recovered in 2013 and 2014 in both Canada and Greenland, fairly disparate locations. A39 emerged in the 1990s in Arctic foxes on Baffin Island and in the area west of Hudson Bay. And by 2000 was circulating in red foxes as far afield as the Northwest Territories and communities in Ontario and Quebec. Now, A3, 10, 11, 12, and 13, it's a little cluster of groups down here we'll just discuss together. Only 5 cases due to A310 were recovered, some from the early 2000s from the Northwest Territories, and 2 cases from the late 2009, from a red fox and a wolf in Kuujjuaq, northern Quebec. A311 and A312 were both single isolates, both from red foxes. One was from Naujut in Nunavut, and another from Churchill in Manitoba. And a single isolate of A313 was recovered in 2002 from a fox in Sinarajak. And let's see if I can spot that one. I'm not spotting it right at the moment, but anyway. And all other isolates of that A313 group. Oh yeah, there it is up there. All other isolates of that A313 group were recovered between 2002 and 2003 down in Newfoundland, which is the outbreak that Hugh was discussing. Now, 14, 15, and 16, the next cluster of groups down here. 14 was found predominantly in Quebec, especially along the eastern Hudson Bay. You can see that in here. In the 2000s, and spread through the red fox population into Labrador by the middle of the decade before disappearing. And group A315 was a single isolate, an Arctic fox in Greenland. And A316 again was just a single isolate, and it was from a little island in Hudson Bay, Sanakiluaq, right in there. And then the 2 groups that predominate in recent years, A317 and A318, which account for quite a large number of cases. They evolved independently from a common progenitor that existed around 2007 from the Times Squirrel phylogeny. And both were first recovered from dogs in different communities in Quebec. And in the subsequent years, from up until 2017, when cases for this study were obtained, both groups were recovered widely across the territories comprising northern Canada, as well as Quebec and Labrador. And A317 is also found in northern Ontario and northern Manitoba. So just looking a little more closely at these 2 groups, we can see that both groups have further diversification within them and fairly well supported subgroups, I guess you'd call them. And so the distribution of those is mapped out on these 2 maps. And what's notable here is that simultaneous circulation of these subgroups and even groups is found in several different areas. So in particular, in the sort of Labrador City region, we have a number of different groups and subgroups represented. So next, we took a closer look, and by we, I mean Susan, at trying to look at the role of the fox species in spreading Arctic rabies virus. So we sort of looked at the distribution of these viral types among host species kind of a little more closely. And so some groups do appear to circulate primarily in an area harboring only one fox species. So for example, A3 2 in Arctic foxes and A37 in red foxes. But many of the samples are in areas sympatric to both, where both fox species are sympatric. So basically, all of the northern coastal areas of the Canadian mainland, Nunavut, a lot of the Northwest Territories, and the area including Quebec and Ontario and Newfoundland and Labrador. And within that vast area, viral groups were typically recovered from both species, with the notable exception of A36, which circulated in the Hudson Bay area, but was only recovered from Arctic foxes. But there were examples within the collection where the same subgroup was found in the same community in the different species, but at similar times. So for example, A38 was found in both species in Tuktoyaktuk in the Northwest Territories and Kugluktuk in Nunavut. And A317 was found in both species at similar times in Umiujak in northern Quebec. So that was a summary of the results from the virus work. And so I'll just quickly run through the fox structure. So we wanted to look at the genetic structure of both species of host. So we had 162 red foxes from Port Hope Simpson, Cartwright, Northwest River, Labrador City, Wabash area, Kuujjuaq, and Churchill. And we had 157 Arctic foxes from Sax Harbor, Igloolik, Baker Lake, Rankin Inlet, Raglan Mine in northern Quebec, and Churchill. And we looked at mitochondrial DNA control region sequences, as well as microsatellites. So two types of genetic marker. So just looking first at the Arctic fox mitochondrial DNA results, basically we found among all those foxes, 26 variable sites, which is not a ton, defining 33 haplotypes. So basically, you have a lot of different sequence types within that set of sequences, but they're only differing from each other by 1 or 2 bases. And so when you look at the relationships of those haplotypes, as you see in this median spanning network, basically you see this just network of haplotypes with sort of relatively little structure to it, no highly supported groups of haplotypes. And when we look at the microsatellites for Arctic foxes, the average diversity was fairly high, but the average differentiation or FST among those localities was quite low. And some of the numbers were significant, but the pattern of it was sort of hard to pick out. So for example, Raglan Mine and Igloolik were significantly differentiated, as were Raglan Mine and Rankin Inlet. Churchill from Baker Lake, but not Churchill from Rankin Inlet, and Churchill from Rankin Inlet as well. So just sort of the patterns were just a little bit difficult to work out. Sax Harbor was not differentiated from anything, but that was only a small sample of size N. So overall, we have low levels of differentiation, mostly or not of them non-significant, and kind of with a difficult pattern. None of the other analyses that we looked at, like structure or principal coordinates analysis or anything that would try to tell you group structure without a priori sample information, detected any differentiation amongst the Arctic fox populations. And this is sort of consistent with the general idea that these foxes do have long distance movements around the circumpolar region and are not highly structured in other studies as well. In red foxes, of course, the story was different. So looking first at the mitochondrial DNA, we had some 130 variable sites, so a lot more variation to deal with, but they defined fewer but more divergent haplotypes. And they clustered into 3 distinct clades: a whole Arctic clade, and then what's called a Nearctic clade, but which includes a mountain subclade, an eastern subclade, and a widespread subclade. And the distribution of these clades among the different localities of fox that we looked at did give us a picture of differentiation of Churchill on the one hand, in the most west, Cartwright in the most east, and then Kuujjuaq, Labrador City, and the other Labrador populations kind of in a separate group. And the average pairwise FST was a little higher, consistent with a greater level of population structure. And structure analysis of the red fox microsatellite data identified 3 clusters as well: a Cartwright cluster, a Churchill cluster, and Port Hope Simpson, Northwest River on the east coast, a little bit different from Labrador and Kuujjuaq, which are the more interior populations. And so we sort of inferred from that we do have this sort of northwest to northeast pattern of differentiation, which sort of suggests the idea of pathways of migration from the north to south, what we call sort of an interior corridor here, and then a coastal corridor here. So that's in eastern Canada, the eastern subarctic region. And I won't discuss that because this is a bit outside this study, but we have looked at this in a bit more detail, and we've got some more detailed patterns to look at going forward. So just to finish off then with some interpretations of all of this, is that the rabies virus phylogeny fits. There are some groups that circulate locally and die off quickly, while others circulate widely over a number of years. We have little population structure in Arctic foxes, consistent with the idea that they do have these frequent long distance movements, and suggesting that they are capable of spreading the virus across this large northern region. And this could, account for this recent emergence and spread of these 2 subgroups. And we have stronger population structure in red foxes as a result of long-term factors and also more limited current movements. But we do have this Churchill versus interior Quebec versus Labrador, interior versus coastal pattern. Cartwright, why it's distinct is possibly because it's isolated coastal region, just a few islands in a bay, or the fox types there may be different because there's been past farming in that area. So there may actually be just a genetically different foxes. They may have, for example, been introduced from PEI. And Cartwright has sort of distinct viral types as well, I think. So anyway, there's something going on there. And so just to conclude, the Arctic fox does play a significant role in maintaining and spreading rabies. Red foxes in northeastern Canada probably function as secondary hosts, spreading the virus southward, and not so much maintain the virus, but contribute to persistence in areas of sympatria with Arctic foxes. And virus lineages do not rely on a single host species to ensure persistence. And this is, if not the first, one of the first studies to apply whole virus genome sequencing with population structure of 2 hosts, and with certainly the hope that it might at least provide some information to help guide future surveillance strategies and feed into other modeling efforts. And yeah, that's it. That's it for me.
Okay. I have a few comments at the end as well. So from my perspective, the point of the research was to try to confirm what pathways or whether we could find anything more on pathway movements. And fundamentally, the hope was based on the fox genetics, since the foxes are much more responsible for the movement than the virus is. As we look at here, and here's an example of 1 year, 2012, and you can see the vast number of different subtypes of virus that exist across the Labrador Peninsula. And if you see in Vujicic, there's the white triangle, Kuujjuaq, and Nain and Makovic. Can we really draw lines between them, or are multiple different foxes further north infected with that subgroup and move down? Similarly, in Nain and Lab City, we see the red triangle, which is also up far north in Cougar. So there's a link, but how far back in time that link exists is really hard to figure out. So I think this is really good initial examination of patterns and leads to more questions. Next slide, please. Looking at that 2012 in Labrador West, just expanding it a little bit, we can see here that Labrador City, Wabush, and Fairmont had 3 different subgroups. So at least three different infected foxes arrived there and started spreading stuff around. It wasn't one fox that arrived and caused the problems. Then you look a little bit further north to Shefferville and Coahuachicamac, and you have 2 completely different subgroups there. So we have 5 different subgroups circulating in the same general area. So certainly there's multiple different foxes moving down, the infected foxes that are spreading this disease. Next slide, please. Now here's 2015, and here we have only 2 different subgroups confirmed on the Labrador Peninsula. In Labrador City, it's all one. It's all the same one, A317B. There might have been other ones around, but we didn't see them. And so you could hypothesize that it was a single fox that was infected that arrived there that just started to spread the virus locally, and a different subgroup that was found in Voisey's Bay and Hopedale. What the link is between the cases in Labrador City and Umiujaq and Senekilluaq, we hypothesize, because the time that these subgroups exist before they evolve into other subgroups, I don't know. So these could circulate for a number of years and not necessarily have a direct line. But it's very interesting to see that some groups we have multiple some years we have multiple subgroups in one area, and other years we only have one. Next, please. And okay, there was one last one there. I don't think I had anything brilliant to say on this one. This is 2017. There was just one subgroup found there. So it doesn't give us a final answer on the questions of the movement patterns. It certainly suggests, as Don was saying, that there is a coastal corridor that the foxes move down. How much they move across the center is still sort of open for me, and I guess research is never over. So that's it for me, and I'd like to thank the researchers for all the work they did in this. I guess we're open for questions now.
Thank you so much for that talk. That was an awesome overview of the history, the ecology, the genetics. So thank you so much. So now for questions. If you have a question, please indicate so in the chat or write your question in the chat, and then we can go through the questions. So who wants to start us off? I have one, actually. I can start it off, if that's okay. So obviously, if there's 2 corridors, how do you think that like future warming is going to affect the movement of these species and the spread of them as well? Because if they're like if it's warming from the south, and then will that stop them from moving downward and then spreading the disease, like to say Newfoundland?
One of the explanations for the lack of pattern between northern Quebec and Labrador is the disruption of where I had the 1 year lag between northern Quebec and Labrador, and how that started getting broken up was it's getting more and more difficult to predict whether we're going to get 4-year outbreaks or whether it's going to be 1 year and then the next year, then a few the next year. And part of this explanation is climate change and how, as the red fox moves further and further north, it'll start to displace the Arctic fox in the Arctic, and how that'll ultimately affect the rabies movement isn't sure. It could end up dying off because the assumption that rabies, fox rabies, the reservoir for it in Canada is the far north. And the assumption is because rabid animals that die, their carcasses stay frozen and the virus stays viable in frozen carcasses across the years until they're scavenged by another animal. But if the temperature warms up such that the virus does not survive as well in carcasses, then it could die off. But these are just different scenarios that people put out, and research would tell us. But there are those who say it'll increase rabies, and those who say it'll decrease it. So there were some written questions. Did you want to add anything, Dawn, to that?
I think that is one of the big questions. It's a really good question, but it's one of the big motivations behind this data to gather. So like our collaborators in ArcticNet, Patrick Layton, Audrey Simon, and I think they're really interested in sort of trying to model that. But yeah, I think it's just unknown. Like red foxes are so adaptable. Like they're found so many places. Like if they were able to move north and displace Arctic foxes, who knows? It could spread more, I would get, or could die off. So yeah, it's one of those things that's just really interesting but unknown.
Thank you. That's super.
The written questions here, one from Kapil, is there any information on virulence or infectivity of subgroups? Maybe somebody like Susan Madden Davis would be able to answer that. I don't know if you have any opinion, Dawn. I'm not aware of any.
No,
and I'm not aware that this has been done experimentally either, where laboratory animals or captive animals have been infected.
Yeah, I'm not aware that it has been done.
And a question from Carla Fury, have we ever seen more than one subgroup in an individual animal? I don't know to what point they look for that, whether they clone out multiple viruses or just take the first virus that appears and then strain type it. So I'm not aware of multiple subgroups coming from a different animal. Again, I think Susan Madden Davis would be able to answer that better. So I could not say that it doesn't happen.
Good question, though.
Yeah.
Yeah, really interesting if it did.
From Alex, have any of the animals that were sampled tagged as part of someone else's study? If so, were you able to get any idea of the migration patterns of specific animals or the movement of specific strains? When I first started thinking about this problem and tried to understand it, this was before genome sequencing was as advanced as it is now. And the only thing I could think of was to radio collar or satellite collar a fox somewhere very far north and hope that it survived and actually moved further south to be followed. It could die somewhere, or it could decide to just stay somewhere. And it was just so incredibly expensive and technically complicated to even try to do that, though I would discuss that theoretically with the university people, it was sort of, this is a huge question to try to answer. Unfortunately, that the genetic typing now had been able to do what radio collaring would have been very limited. So I'm unaware of any of these animals being tagged. Most of them came from either trappers that were paid for the carcass after they had skinned them, or they were from the DNA of brains that were submitted to CFIA for rabies confirmation. So there would not have been much additional information to that other than place and date. Is there any evidence of recombination between the subgroups? That's a difficult question, so I'll pass that one to Dawn.
It's not something I'm aware of. I don't know enough about how variable these sequences are, honestly, to say anything more about it. But certainly it's not something that Susan has mentioned, that could be occurring. So that would, I guess, also be if a host had two different types, is when recombination might occur. So that's a very good, interesting question. But I don't know that there is, yeah. With regards to Alex, your question, the only tagging sort of that we know of was that the Arctic fox that traveled like 42.6 kilometers, or that was in the news recently. Wasn't part of our study, but I mean, that could be a really good thing to do. And I've been wondering too, like if we just went in with like a really detailed, like whole genome kind of analysis of individual foxes, would that give us any useful information? Sort of like, as Hugh said, when we started this study, it was kind of back before like RAD-seq or genotyping by sequencing, these kinds of techniques. I think it could be a good approach to just take some of these foxes and look at really how closely related are they, but that's wasn't part of this study.
Are there any more questions that people want to ask, either in person or in the chat?
There's a question just popped up. Is there a significant difference in the expression patterns of the different rabies strains, barring species differences? It's possible. It's possible that there was I don't know if I'm going out on a limb on this one. There's very few cases of people surviving rabies, and very few cases where treatment actually worked. There was a young lady in the US who was bitten by a bat who was treated with what's called the Milwaukee Protocol, and she survived. And the suggestion was that it was only a weak bat strain. And so it's possible that expression varies. Certainly in humans, any exposure is considered to be identical in that they all receive the same post-exposure treatment, and regardless of whether it's assumed that this is a case of bat rabies in a fox, and therefore maybe the fox is not expressing virus in its salivary glands. The treatment is universally the same, because academic questions are not practical questions when it comes to treating people. There is, we refer to two different types of behavior, aggressive and passive, in animals that are affected with rabies, and whether this is a reflection of the strain that they are infected with or not is a good question. the actual, as far as I'm aware, research into infecting experimental animals with rabies, if you see films or videos of rabies behavior of rabid animals, they tend to be fairly old. These were done in the 60s and 70s, and the days of just infecting somebody's dog or cat, or a dog or cat with rabies virus just to watch its behavior, those days are kind of slipped by. There's much more control over why you do something, the stress or impact on the experimental animal. So to break this out and do it experimentally, I guess theoretically is possible, but practically I'm unaware of it happening. I don't know if others have other answers for that.
Some really great questions that we had today. I think that speaks to how interesting the talk was. So if I could say, welcome and thank you again for agreeing to do this seminar. And, we've been trying to do this since March, so I'm really glad it finally got to happen. Thank you so much.
Thank you for your interest.
Have a great day.
Yeah, thanks, everybody. Nice to see some faces.
Over and out.
Thank you. Bye.
Bye, Carla.